Good morning, everyone, and welcome to this year's Crafoord Symposium. My name is Göran Hansson. I'm the Secretary General of the Royal Swedish Academy of Sciences, and I'm delighted to see so many of you here with us today to celebrate the Crafoord Prize, this year's Crafoord Laureate, Professor Chisholm, and to learn more about marine microbiology. But before we go into the program, I thought I'd tell you a little bit about the prize and how it all started. And it all started, well, to give you the prehistory, it started here in Lund in 1949 or 50, when there was a young physician-scientist by the name of Nils Alvall, who had a patient with kidney failure. And Alvall had been playing with trying to construct an artificial kidney, so he decided to use that machine. And somehow it worked, and he could save the life of this young fellow who was his patient. He developed this further. There was another scientist in the Netherlands, Wilhelm Kolff, who was on a similar track, so they were parallel to each other. But Alvall had the fortune of teaming up with a businessman and entrepreneur, Holger Crafoord. Crafoord had made a lot of money from being involved with the company Tetra Pak that makes packages around milk and things like that. And he decided to move all his energy and skills into the artificial kidney. So he had a company that he owned, a very small one called Gamla Brogatans läkemedelsaffär, tror jag den hette. I think it was called, and with a short name Gambro. And they developed that into a manufacturing company for dialysis machines for artificial kidneys. That became a world leader and contributed to saving the lives of literally hundreds of thousands, if not millions of people around the world. Many years later, they merged with an American company called Baxter, and that so often happens when a European company merges with an American one. The company is now called Baxter. So the Swedish name disappeared, but nevertheless, it started in here and in the way I described. Holger Crafoord decided to use quite a lot of the money he had made from Gambro to set up a foundation to support research. And he decided to focus on areas not covered by Nobel Prizes, but still extremely important for mankind. And it was decided to set it up in a cycle. So it's awarded in mathematics and astronomy one year, geosciences the next, biosciences the following year, and then a fourth year in polyarthritis, rheumatoid arthritis, essentially, if there is reasons to award a prize in that area. And the reason for that more focused prize is that Holger Crafoord himself suffered from that disease and was severely crippled. That prize has been used to award major breakthroughs in immunology that has led to clinical benefit in arthritis. Last year, the prize went to geosciences and to Susan Solomon, who has done fantastic work on atmospheric chemistry and the ozone hole and how to heal it. The prize or invitation to nominate for the prize are sent out by the Royal Swedish Academy of Sciences that handles the whole prize award process. So we send it out to scientists all around the world. We get more than 100 nominations back every year. And then the prize committee works hard to identify a shortlist, and in intense discussions within the committee and with members of the academy and with experts around the world, they finally focus on the individual scientists that has made the most fundamental, most paradigm-shifting discovery worthy of the prize. And over the years, the prize in biological sciences have gone to people like Edward Wilson and Carl Woese. Last time, 2015, to Tomoko Ota and Richard Lewontin. So that's the story around the prize, and I shouldn't take up more of the time. I know we're all excited to hear Dr. Chisholm's talk and learn more about Prochlorococcus and the rest of the marine biological universe. So I'll stop there and ask Dr. Daniel Conley to give us some instructions in case there is a fire. But stay tuned for the rest of the symposium.
Good morning. Crafoord Laureate, Professor Chisholm, members of the Royal Swedish Academy of Science, and ladies and gentlemen. I also warmly welcome you to this amazing symposium on how microbes rule the world. I'm expecting lots of heated discussion, but no fires. But in case there is need to leave the room, there are 2 exits at the top, and there's an exit over here on the side. We'll have a coffee break following the first lecture by Professor Chisholm and questions, and that'll be outside. And we also have a lunch for registered members outside as well. And I'll be the moderator for this section. I am a professor of biogeochemistry in the Department of Geology here at Lund University. And with that, I'm going to turn it over to Professor Jarone Pinhassi, who's a member of the Swedish Academy of Sciences in the Biosciences class, and he will introduce Professor Chisholm. So, Göran.
Thank you, Daniel. Crafoord Laureate, Professor Chisholm, members of the Royal Swedish Academy of Sciences, ladies and gentlemen. It's a great pleasure to introduce Professor Chisholm to today's lecture and today's symposium. And we heard yesterday a bit about your history, and I'm going to repeat a little bit of it. With a PhD in '74 in upstate New York, is it? And I noticed the first research publication, might be wrong, but causes of daily rhythms in photosynthetic rates of phytoplankton. And you emphasized yesterday that you have been working on growth of phytoplankton throughout your academic life. And following the PhD, went for a postdoc at Scripps, and which is one of the major institutes still globally in marine sciences and in marine microbiology in particular, maybe. And after that, moved to MIT, where you got the prestigious nomination for Institute Professor in 2015, I think, as a sole women so far, or is it? There have been a few. That's nice to hear. I was wondering. Okay. And then, so I would like to emphasize that research on growth of phytoplankton, for example, diatoms and also some green algae, and how environmental factors regulate cell division was a focus, which, as you mentioned yesterday, led you on to using flow cytometry to see how do microbes do, how do phytoplankton grow. And that led to the discovery of Prochlorococcus in 1988 when the paper came out. So we would be curious to know when was the first hunch you had that what was the year when you first guessed that there are some organisms out there that are these organisms. And interestingly, after that discovery, there is a series of papers on Synococcus, and I guess it's not that they are misidentified. It's actually a true interest in this also cyanobacterium that is incredibly important in more maybe coastal oceans. And I noticed also that until 1998, a decade later almost, the grand majority relates to publications that on phytoplankton rather than Prochlorococcus. Probably reflecting, of course, a highly active research activity in your lab, showing, I mean, that you just can't let go of course. But and since '98, this research on phytoplankton in general and on Prochlorococcus in particular, I think has stimulated and inspired research in a grand variety of research fields, not only marine microbiology, but also, I mean, the groundbreaking findings and the methodologies and ways to do research has inspired in molecular biology, environmental sciences, and a wider chemistry. And maybe most of all, or in addition, not least important, the public curiosity in what happens in the oceans and how our ocean functions regulated. And with that, we greatly look forward to, you were teasing us a little bit yesterday with all this kind of new research, so we look forward greatly to hearing your talk today. Thanks.
Thank you. Thank you so much. It's interesting to see your whole career laid out like that. And I hope I didn't set you up too much yesterday for disappointment today, but I'm going to fill in some of the blanks from yesterday. First, I just have to say how thrilling it is to be here, to have been awarded this prize, and for me personally, obviously, but for our field, I think you know, this is our time, and to have it be recognized, I think, is particularly wonderful. So let me tell you a little bit more about Prochlorococcus. This is the outline today, and I have way too much to tell you. I'll talk about generally the biosciences, which we hope to change with our approach to microbes. Prochlorococcus, I'll fill in some of the blanks, and then I'll end with a bunch of new mysteries that we have. I probably have more questions than answers, but I hope it'll stimulate discussion. First, the biosciences, and I'm preaching to the choir here. I know you're all thinking the broad scale, but I always start with this, especially at MIT, where much of the work in biology is on the how the machinery works, and I argue that we really need to know this level in order to understand the why, and the why of life on Earth really influences the how. It's all one big system. And as I argued yesterday, microbes, and for me, Prochlorococcus in particular, is a beautiful system for studying life across all of these scales, from the molecular to the global scale. And properties emerge that you wouldn't see if you just studied life at a particular single scale. So here's Prochlorococcus, the new love of my life, besides my husband. And you can see them fluorescing red there in the background. They're a cyanobacterium. They have chlorophyll B. They're less than a micron in diameter. They are widespread through this region of the Earth. They don't like the high latitudes. We're not sure why. There are 10 to the 8 cells per liter in most places, and 3 times 10 to the 7th globally, which is 2 times the weight of the human population. So there are a lot of them. And sometimes I define them as the smallest cell in the ocean that fluoresces red on a flow cytometry signature, because that is them, unambiguously. If you find that little spot and sort it, those are Prochlorococcus. So they're amazingly stable. This is a time series off of Bermuda, showing cell abundance seasonally up and down. This is a very old data set. We have newer ones that go much longer. And this is its cousin, Synechococcus, which we did study for years. In fact, we set out to study Synechococcus, the flow cytometer, and Prochlorococcus appeared. And I added this. Synechococcus feels slighted by me because I moved on, and it's very important in the oceans. And it's because it's a little bigger, it's less abundant. But in terms of biomass, it's probably more, carries more weight, and it's more widespread. It's very important, and don't neglect it. Okay. But I'll move on to my favorite, because it's smaller and simpler, and we like it. So we learned, as I said yesterday, early on, that Prochlorococcus is not one thing. It has ecotypes. And what I learned yesterday is the ecotype concept actually originated in Lund. And I've forgotten the name of the person, but it was relative to plant ecology, and we have adopted it here, showing these are different strains of Prochlorococcus that have different light optima for growth. These are high light adapted, and these are low light adapted, and we were able to develop probes so that we could look at them in the ocean, and you can see that it makes sense that the high light adapted dominate the surface and the low light adapted are more abundant in the deep water. And if you take these two high light adapted ones and you look at their temperature adaptation, you see that they have different temperature optima curves. And you go to the ocean and map their distribution, and evolution works. They are distributed with the low temperature are more abundant at the high latitudes, and the high temperature adapted ones are more abundant here along the equator. So this was before genomics. This was when we were just studying them physiologically, and every time we'd isolate a new strain, we'd see whether they fit into this pattern. And here's the same time series, looking at breaking down these total numbers into the oscillations of these different ecotypes. And you can see, if you look closely, that they're out of phase. So as the environment changes, their relative abundance changes, keeping the whole population relatively stable. There's even one here that we've determined is adapted to the deep mixing, the fluctuating light. It does better in fluctuating light than some of the others. Okay, moving on to genomics. And I'm finally showing a phylogenetic tree. Yesterday somebody asked for one. I try to avoid them, but here they are. Here's these highlight adapted ones. They're a very tight cluster, and then the low light ones are spread out. So as we started being able to sequence the genomes of all of these strains that we have in our collection, we found that they're dramatically different. This is the genome size. Each one of these is one of the different colored we color the ecotypes to keep track of them, and I drive my students crazy making them keep the color code through the years, and it's really helpful to me as I make slides. Otherwise, I lose track. So you can see dramatically different genome sizes. The low light adapted are much larger, and they have dramatically different GC content in their composition. So they're really quite significantly different. So as we started getting more and more genomes, this is the image in my head of the oceans. It only has Prochlorococcus. And just started wondering, a milliliter of seawater, how many different Prochlorococcus are there? And how many different Prochlorococcus are there in the global oceans? And that's my goal before I retire, so I have to live quite a long time. But we're getting closer, so let me fast forward to very recently a massively new database, thanks to the Simons Foundation that funded. Another one of my goals was to take all the samples out of our freezer and have them sequenced. And so there are strain genomes, metagenomes from all over the ocean, and single-cell genomes, thanks to my collaborator, Ramunis, who we'll talk later. And also thanks to Paul Berube and Steve Biller in my lab, who stewarded this whole thing, which it's easy for me to say, take these things out of the freezer and sequence them. And then it's very hard to get them all processed and get all this done. So they just published these 2 papers with all the data and scientific data, which is there, and I hope to inspire anybody to go use and plow through these data, because there's all kinds of treasures in there that we don't have time to look at. So here's a phylogenetic tree of all of those. It's a whole genome tree, basically, of all of those sequences. And here are the high light adapted and the low light adapted ecotype clades, we call them. And if you look at the GC content, you can see how dramatically different it is as we go down to the low light adapted. And this is the percent genome coverage, because some of these are single cells. You can't get a whole genome from a single cell. But Ramunis' skills came close to getting a significant fraction of the genome for some of these. So where the blue is already all the way to the edge, the 100% are the strains we have that we use as reference genomes. And here is the genome content. So each row is a genome, and these are the core genes that they all share. Some of them are absent because they're not complete genomes. And these are all the flexible genes that are distributed among the broader population. So the big question is, what are these and what are they doing? And so if we look at the pangenome, which is what we were just viewing, in another way, this is the number of genes and the number of genomes sequenced. Every time we sequence a new genome, we see about 100 to 200 new genes that we haven't seen before. And you can do a theoretical analysis and project this, which takes you to 80,000 genes projected in this total Prochlorococcus collective, which is 4 times the size of the human genome. And as I said yesterday, 20,000 of these we already have in our hands, in our collection. And about 1,200 of those are core and 20,000 flexible. And these, most of them are of known function, but these are of unknown function. And that's where the mysteries lie in the Prochlorococcus Federation, as we call it. So with this genetic roadmap, I call Prochlorococcus an aspiring finch because it gives us clues, just like the finches. This is not to analogize me to Darwin, but the finches gave him clues about the selective pressures from their beak sizes. Prochlorococcus gives us clues about the oceans from its gene content. And so I'm going to tell you just a few of the stories that we've unraveled here. Somebody asked a question yesterday, so here's an answer to that. This is Shane Hogel, a postdoc in my lab, did this analysis. This is the siderophore transporter gene content of Prochlorococcus, and you can see that there are none in the samples in the Atlantic and some in the samples in the Pacific. This is sort of a there or not there analysis. And if you look at the iron concentration in the 2 oceans, you can see that it is higher in the Atlantic, and that's why these Prochlorococcus gave up having that gene. They said, we don't need it. There's a lot of iron here. That's what they tell me. I talk to them. And then you look at phosphorus. This is, it's much lower in the Atlantic than the Pacific. And if you look at Prochlorococcus genomes of some really closely related Prochlorococcus, so each one of these is a genome, highlight 2 clade genome. And you can see here that these Pacific cells don't have any genes in this region. I mean, they don't have these genes. And all of these genes in the Atlantic, and this what we call a cassette, are phosphorus acquisition genes. So if you compare 2 very closely related cells from these 2 oceans, they tell you what they're experiencing. And my big question is, what about all these genes that we don't know the function for? And I'm hoping that people will take this data set and look for patterns in the genes of unknown function that are screaming to tell us a story. If you can see that they're distributed differently along environmental gradients, it means that those genes are being selected for. Then we have to figure out what their function is, which is one of the biggest challenges in our field, I think, right now. There are so many stories we're missing because we can only look at the things we already know something about. So I think that's an exciting frontier. So the emerging image here, then, of this Prochlorococcus Federation, as we call it, or collective, is that you have light and temperature adaptation are very deeply rooted characteristics, and then superimposed on that, you have this variability in these gene cassettes. And there was this beautiful image in the New Scientist of how microbes are constructed now, with this Rubik's cube of combinations of their gene composition. And so the way we see it is that in different environments, select for different combinations of these flexible genes, which is what contributes to the stability of these populations. So here's the slide I showed you earlier. Now this is my image of what's going on in these within these ecotypes. There's all of this. We had when I made this slide, we had about 5 data points on these, and luckily, Jed Furman with Nathan Algren, who actually was also in my introductory ecology class at MIT many years ago. He's now a professor at Clark University. Just published this paper of Prochlorococcus at a time series off California. And these are the abundances of different RNA ITS sequences of the different ecotypes. So this is the real data that shows how this fine scale diversity really changes with changes in the environment. Okay, so where does this diversity come from? What is the mechanism of all this genetic exchange? And this is a big mystery because we've been trying for 15 years to genetically modify Prochlorococcus in the lab and have been unable till very recently to get it to even take up DNA, let alone incorporate it. So, so we know transformation, these are the typical ways that bacteria move genes around, and doesn't appear to occur in Prochlorococcus, nor does conjugation. We've tried that over and over and over. Transduction by phage, there is some good evidence that that goes on, but it doesn't seem to be enough to account for the diversity that we're seeing. So the story we're working on, if you look at a position along a Prochlorococcus reference genome, and you recruit all of these genes from all of these genomes that we have, you see that, as you'd expect, the core gene, the number of genes with the number of genomes with a gene, so the core genes have the same frequency in all the genomes as you'd expect because they all have it, but there are these islands of variability where the flexible genes are concentrated. Like the blue ones are the flexible genes. And so they're abundant, their relative abundance is all over the place because every cell doesn't have them. And recently, Thomas Haeckel and Marcus Ankenbren in my lab dug into this database very carefully, as only these skilled young bioinformaticians can, and really looked for all these islands areas in all the genomes. And so these are, each one of these is a genome, and the gray things are island regions, and they've lined them all up according to the position along the genome. And what they found was that they had some very uniform characteristics. And for example, they have this transfer RNA on one of the ends. They have an integrase gene, which is involved in integration of DNA into genomes. They have these what we're calling integrative elements that are identifiable, and I'll talk about those in a minute. And a lot of the areas are involved in phage defense. So the hypothesis that we're working on, and we have some good evidence for this, is that these integrative elements are what are moving components of the genome from one cell to another, or from one part of the genome in a cell or another. And this integrative element here, they include either nutrient acquisition genes. For example, one we found is the complete nitrate assimilation pathway genes that is in one of these elements. They have phage defense systems, which I don't have time to go into, but they're called pickies. They're really neat little system. They're like phage that infect phage, so they kill the phage. It's really cool. We don't have biological evidence for it, but in other systems they do, and we think that's going on here. And stress response genes. And so what Raphael Lorenzo, a postdoc in the lab now, has shown is that when you put the cells under stress, these integrase genes are upregulated, and the element circularizes and pops out of the genome. It replicates, and it inserts itself in another place, either in the cell or in another genome, which I'm going to try to convince you actually happens in a minute. So how do they move? And this is what I was setting up yesterday. So I hope I convince you. And yesterday I talked about these vesicles that Prochlorococcus makes. Steve Biller, postdoc in my lab, worked out this system several years ago, showing that Prochlorococcus buds these little lipid-bound vesicles, and they include DNA and proteins and active enzymes and all kinds of things. And they're very abundant in the oceans. And they're produced by diverse bacteria in the oceans. Because they contain DNA, you can sequence their metagenome and identify what bacterium popped out that vesicle. So what we did was sequence the metagenomes of Prochlorococcus vesicles from the wild, from Dave Carl's station, the hot station. We're very indebted to Dave for his generosity in making this station and the cruises available so that you can go out to Hawaii and go out in the water and collect samples for a few days and do these kinds of analyses. And so this is a metagenome from samples at the hot station. And this is the relative gene copy number in the cells. And this is the relative gene copy number in the vesicles. And you can see that the core genes are equally represented in the cells and in the vesicles. But these integrase genes and these regulation and integrative element replication genes are all overrepresented in the vesicle fraction. So you say, oh, well, okay, that's one sample. They screwed up the metagenome or something, which is what I said to them, because it's too good to be true. But look at this. So these are different cruises and different depths, and you see that every time that the vesicles are enriched with these little elements. So we think that that this is real, and we think it's very exciting, because now I have this image of these vesicles sailing around in the ocean and looking for another cell to pop into, and that not only among Prochlorococcus but even with other microbes, that this is the sort of the genetic marketplace of this large metabolic superorganism is what I think of it now. Anyway, so Thomas, we're still discussing this. Thomas wants to call them integrative and replicative vesicle encapsulated elements, ERVs. But there may be other ways that these elements move. So this is just a hypothesis at this point. But this is the way we see it happening, is that here's the cell with an island. You have some kind of stress that makes this pop out into the vesicles and find another cell and pop in. And this is a way to generate diversity within the community as the stressful situation changes the environment, so that you have a pool of different genotypes to draw upon to contribute to this stability. So the question is, does this really happen? And we don't know yet, but I can give you some evidence. Steve Biller and Aldo Erlano in the lab did this experiment where they stained the vesicles with fluorescent dyes from different cells. And the first question is simply, do they stick to other cells? You have to stick in order to deliver your cargo. And the answer is yes, they did. They were able to measure by flow cytometry where those fluorescent vesicles, what they stuck to. And then you say, well, everything's sticky. Everything sticks to everything. And so do they discriminate from one cell to another? And luckily they do. So these are vesicles produced by these different strains, and these are the recipient strains, and you can see that some of them do not stick to those recipient strains. So this is just a work in progress. Do they deliver their cargo to the other strain? Steve is working on that now. We hope so. Otherwise, this theory is not sound. But so far, this is looking like it might be an important process in the oceans. Okay. So there are so many mysteries that I want to tell you about, and so little. This is me channeling Prochlorococcus. this is how we work. We let the organism tell us what to do next. I always tell people I don't really have to have ideas because Prochlorococcus gives them to us. They're always doing something weird that we have to figure out. So I want to tell you just some of those stories. And most of them don't have an answer. So I'm throwing them out, but they're at least a good solid question. So yesterday I showed this slide showing the thin film of phytoplankton on the surface of the ocean, and that when the wind blows, this mixes, and some of the cells get mixed down to the deep ocean, and this one went rogue since yesterday, but I left it because I kind of think it's kind of cute. And that's probably the way it works in the ocean, too. So our question was, how long can they stay alive in the dark? They're photosynthetic. So we played around with them and did some experiments. This is Alice and Co. and Steve Biller in my lab, where you take the cells and you grow them on a light-dark cycle, and then you put them in the dark for different durations, and then you see how long it takes them to survive and see if they survive. And to make a long story short, they survive for less than a day if they're in pure culture. If you put them with one of their friends, one of the strains that they were co-isolated with, that we spent years getting rid of so we could do pure culture experiments, but you put them back with a pure strain of, in this case, Ultramonas, which is one of its favorites. It's a heterotrophic microbe. They can last for much longer in the dark. So the experiments. So then we asked, well, what if you expose them repeatedly to darkness? Do they get better at surviving? And the answer is yes, which was really surprising to us. So here's an exposure to 3 days in the dark. It takes them 24 days to recover from that. Then you expose them again. It takes them 5 days to recover. You expose them again. It takes them 2 days to recover. You do it again and again. It's 2 days, 2 days, 2 days. You put them in the constant light-dark cycle with no dark stimulus, no extended dark stimulus. Just let them grow, 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 grow. Expose them again, 2 days to recover. They've learned. And so, moreover, those, what we call them the trained cells, survive longer in the dark period than the untrained cells. So something has changed. And we've sequenced, now you can do these experiments, you've sequenced the genome here, and there's no genetic difference between the two populations. So what is the mechanism? And I don't have an answer, but there's obviously some epigenetic phenomena going on here. So again, this organism is dragging me into a field I know nothing about, and we're starting to work on what that is, and we think there might be some really interesting regulatory phenomena or DNA methylation or all those kinds of things. So we're working on that. Another thing, okay, so these are the two most abundant microbes in the ocean, Prochlorococcus and SAR11, which is Pelagibacter, even smaller than Prochlorococcus, a heterotroph with a tiny genome and a very small cell. And I had a postdoc, Jamie Becker. I said, these are the two most abundant microbes on Earth, and they live together in the ocean. We should be able to grow them together and understand how they talk to each other. And he took it on, and it took him 2 years to get the media to get them to grow together. It was a labor of love, and I admire him for doing that. So here is the Coculture of 2 Prochlorococcus strains with SAR11, and you just serially diluting, and Prochlorococcus is feeding SAR11, so they're following each other. So we're able to do some experiments now to try to get at the conversation between these two incredibly tiny and important microbes. And I'll just show you one. Jamie just published this paper that I say they're not always the best of friends in co-culture, batch culture. This is Prochlorococcus and SAR11 growing together. Prochlorococcus doesn't care if SAR11 is with it. It does this with or without it. But it feeds it, and SAR11 grows and grows. But when Prochlorococcus reaches stationary phase, SAR11 says, nope, don't like it. We don't know why. We're going to figure that out. But what's interesting is that if you grow Prochlorococcus with another heterotroph, which is what we call a copiotroph. It likes high concentrations of carbon. You get a completely different response. This one likes it when Prochlorococcus goes into stationary phase and has a little boost in its growth. So now we're trying to compare the metabolites in these two cultures to see what's going on with that exchange. Stay tuned. Another interesting feature of Prochlorococcus that I alluded to yesterday is that I forget who. How's my time? I guess I'm okay.
Yeah, 10 more. Yeah, 10.
Oh, dynamite. Okay. I've been waiting for the flash here. It's 10. Okay, or the hook. Kind of. Okay, so another interesting feature of Prochlorococcus that I alluded to yesterday is that here's the assimilation pathway for nitrate in typical organisms. They assimilate it, they reduce it to nitrite, to ammonia, and amino acids, and that's their nitrogen source. As I alluded to yesterday, some Prochlorococcus don't have this pathway. They don't have this gene. And it's very scattered. Here's another phylogenetic tree of the low-light adapted Prochlorococcus, showing that some of them don't have it at all. And some of the clades that have it, just some cells have it and others don't. And among the highlight adapted, it's completely scattered around, plus or minus. So this is the work of Paul Berube in my lab. I've been working on it for quite a while. And what Paul noticed was this is a depth profile off Bermuda. And these are the low-light Prochlorococcus, the total low-light Prochlorococcus that have a peak down here right near what's called the nitrite maximum layer in the oceans. But only a fraction of those have the nitrate reductase. They don't all have it. And so the cells that can reduce nitrate to nitrite in this region, because there's not a lot of light and they don't have a lot of energy, actually excrete some of that nitrate. And we've shown this in the lab, that they take it up, but they excrete it. And so our hypothesis is that these guys are taking it up, excreting nitrate, and the ones that can't produce nitrate, don't have nitrate reductase, are taking that up and using it as a nitrogen source. And we have some evidence from our culture work that this is going on. So I love this because I think of them as partners and working as a team out there. And it'd be really cool if this is one of the ways that they share their genomes physiologically. Another story that's emerging that we're pretty excited about is that these low-light 4 Prochlorococcus encode a pathway for degrading chitin, the invertebrate shells. And we don't know why would Prochlorococcus do this, and we still don't know why. But here are the genes in the pathway, and some of them don't have the initial chitinases. That this is the way the pathway operates. We think chitinase is extracellular to start degrading it so that the rest of the enzymes can kick in. And so one of the questions is, do they degrade it? And we have some evidence that they do. And it turns out, and if you ask why, we think that maybe these low-light 4 are sticking to particles, chitin particles in the system. And this is just a picture of the bottom of some culture tubes comparing a highlight strain with a low-light strain. And the lore in the lab was always that these low-light 4s are sticky. You know, they settle and they make these clots and everything. And it turns out they also have cadherin-like domains, which are things that make cells adhere to surfaces. So this is a new story that's emerging, that Prochlorococcus may be hitching a ride on these particles and using them either for carbon or nitrogen. We're not sure, and we're working on that now, but it's kind of a new dimension. The other thing that's pretty cool about this story, and this is from a colleague, Greg Fournier, in EAPS, who works on molecular evolution, is that this is where we think there was horizontal gene transfer of this chitinase gene into Prochlorococcus because other cyanobacteria don't have it. And this is exactly when, he tells me, that marine arthropods diversified extensively. So it's so cool when you see evolution works. So there are all these arthropods around, and Prochlorococcus said, boy, there's something there to take advantage of. And they grabbed a gene from somewhere. And off they went. Okay. So I also talked a little bit about this yesterday. Prochloricins, which are secondary metabolites that Prochlorococcus makes in huge abundance relative to any other type of bacteria. And where other bacteria make 1 or 2 of these. Nisin is an example that's used, as I said yesterday, for preservation of dairy products. It's an antimicrobial. And so we're trying to figure out, and these guys encode maybe 20, Synechococcus up to 80. I hate it when Synechococcus is better than Prochlorococcus, but so I don't always mention that, but I have to this crowd. But they both make a lot. And we're trying to figure out what they are, because they have these tiny genomes. So, why would they give so much area up to these genes unless they're important? So what is their function? And I don't have an answer, but we have some hypotheses. One is that they're some kind of signaling compound or antibiotic, and Daniel Scher from my lab and his student published a paper a few years ago showing that they're at least upregulated when you add another microbe to the system. The other theory that's floating around in my lab, Raphael Laurenceau, is suggesting that they're defense molecules, because there's evidence that these types of molecules in other systems are protease inhibitors, and actually microcystin from some freshwater cyanobacterium inhibits grazing in Daphnia or something. So we think that might be a way to survive ingestion by little protists, and we're starting to work on that. The other theory is that they're part of the metabolic marketplace and the currency between organisms, because the students noticed that prochloricins are very enriched in reduced sulfur and glycine. Here's a structure with these sulfur bonds. And Pelagibacter, which is SAR11, the little cell that I showed you in the Coculture, requires reduced sulfur and glycine. And we actually have evidence for this one. So here's SAR11 growing with one of the prochloricins. And if you get rid of the sulfur bonds and you can make a linear prochloricin. This is in collaboration with our chemist Wilfred van der Donk, who's been very excited about Prochlorococcus making these things. He's a chemist. He loves their structure and all of that. And he made linear versions for us, and you can see that SAR11 does not grow well without the one with the sulfur bonds. So we think that there's something to this, and we're continuing to work on it. Okay, just to wrap up, this is a review article that Steve Biller and Paul Berube wrote a few years ago. And when we called it the Bacterial Federation, and I always get a kick out of this because the artist has them marching on land. These are all the different ecotypes with their flags marching on land. But I like to show it. And but when I look at it, I think, what if we'd isolated a single Prochlorococcus, and I'd spent the last 25 years working on that strain? I would have a completely wrong image of how the oceans worked. I would have, it just would have been so distorted. So I think of all the model bacterium that we used to build the biological sciences knowledge that we have today, which has been, extraordinary breakthroughs. There's no question about that. But what are we missing? And I talked to my colleagues in the biology department, and I say, you really should add another organism and see what happens. And we have great conversations. And so I think we we all know this, but we can celebrate ourselves. I think we're on the frontier, and even though sometimes it doesn't feel that way. But I think we are the frontier of the biological sciences, and that slowly we're going to get more tools and really be able to understand these systems. We're just starting to scratch the surface. And I love this quote from Nigel Goldenfeld and Carl Woese, who was one of the recipients of this prize. And I'll talk a little bit more about that tomorrow when I accept the speech. He's one of my when I accept the speech, when I give the speech to accept the medal, because he was one of my heroes, and I just love this this image of emerging picture of microbes as gene swapping collectives demands a revision of organism species and evolution itself, and I think that's where we are, and we're the ones that can do that. So. With that, I have to think that this. I think the majority of the people, I probably missed some people that have contributed over the years to this story. There's Stefan, right there, Bertelsen. There's other people in the room too, but and I can't go through everybody and who did what, but I do need to highlight. First of all, Rob Olson, who was the key to the discovery of Prochlorococcus. I mean, he's the guy that got it, that first built the first instrument, got it on the ship, and saw the noise that we thought was noise, that eventually was not noise, was Prochlorococcus. And this is Paul Berube and Steve Biller, who are in the last decade the guys that really drove this, massive expansion of the genomes and the new directions we've been going on. And this is Alison Coe, who's been my lab manager for years, and she's the magician that can keep all of these cultures going and keep everything happy, and also does these incredible experiments of the dark adaptation that take years to get answers to, because she's just fascinated. She's at one with Prochlorococcus. So with that, the tremendous gratitude to all the agencies and people that have funded our work, and of course for this incredible opportunity to share it and all of it. Thank you.
Thank you for another fantastic seminar. This is just wonderful. We have time for some questions. Way back in the back. I want to note that next to at the end of each rows, there's a microphone, and there's also a person walking around with a microphone.
Press the button.
Okay. Being a diatom person and preferring to work in polar areas, can you hear me?
Yes.
Yeah. The obvious question from me, what do you think is the potential for Prochlorococcus to go polar?
That's a great question. We don't know why it doesn't. There's several theories, whether it's just temperature or it could be that there are more nutrients in the high latitudes, and the Prochlorococcus doesn't compete well at high nutrients because it specializes in low nutrients. So that's one factor. But as I talked about yesterday, as the oceans warm, they will expand, but probably never. They will never take over the high nutrient areas, simply because that's not their specialty. Their specialty is being the smallest, having the highest surface to volume ratio, and being able to eke out a living where others don't do quite as well.
Okay, Kirsten?
Okay, so the question from me this
there
time
you are.
is that I want to remind us about 4 years ago, the Crafoord Prize went to Tomoko Ota for the near neutral theory.
Oh.
And I assume that you grew up, as me, under the paradigm of everything being neutral or almost neutral. So now, I want to ask you, how many neutral genes do you find in Prochlorococcus? Or is there any neutral gene at all?
Oh, that's a good question, and I absolutely don't have an answer. But I would imagine, I mean, one of the things that frustrates me is that we study the things that we know we're going to find some variability. Like we knew phosphorus was a selective agent. We know iron is, I mean, now we know it, because we have these global measurements. So then we go to the genomes and say, what is that, But I imagine among all these flexible genes, there are many that are neutral, and the core genes that they all share. But I must say that I'm not at all an expert on we kind of steer away from population genetics, which we shouldn't, but I get so confused when I get and so, but that's a really good question, and we haven't really dug into it.
Thank you again for a wonderful lecture. So, as a phytoplankton ecologist, I struggle with growing things in the lab, and I don't know how easy or difficult it is with Prochlorococcus. But I'm wondering, to what extent do you think that by sequencing all the genomes and exploring those, will that help us culture more strains and species, do you think?
That's an interesting question. Yeah, I can give you one example. Actually, I have two answers because I can explain that gap of working on Synechococcus after we discovered Prochlorococcus, because we couldn't get them into culture. So you had to go on a cruise to study them. But we had Synechococcus, so that's why there was a 10-year gap. So they were difficult to get into culture. And I have to credit Brian Palaniuk, who was working on Synechococcus in my colleague's lab right next door, and he took it on and he got them into culture. But to answer your question specifically, this nitrate reductase story is really interesting. I was tempted to give you the whole 20-minute version of that because it is such a journey. Because when we first isolated Prochlorococcus, we had media with ammonia, so we'd isolate them, and then they wouldn't grow on nitrate. And that was heresy. Everything in the ocean is supposed to use nitrate. And we went, huh? But we didn't have genomes then. So then we sequenced the genome, and they didn't have the nitrate reductase gene. Oh, science works. So then we said, but this can't be true. So so we went out, and Adam Martini, and I got to make sure I get all these people right, a group of students and Adam, Paul Berube was involved in that, went to the GOS database and were able to find segments of genomes that were clearly Prochlorococcus that had the nitrate reductase gene. So we said, so they're out there. So then they went out and they made media with only nitrate and selected for those strains. So there's the only example I can tell you, but so it makes a difference. If you know from a genome that they have that gene, you can design your media to get that strain, and that's how we've got now lots of strains in culture that can use nitrate. And the universe is in order a little bit more, but there's still a lot out there that can't.
Okay, over on this side, we'll take.
I have a question about the training of the cells to survive in darkness. That was a quite impressive period of time. So I'm wondering, are cell divisions happening during that extended period, or is it really the same cells all along?
Yes, I went through that rather quickly, but and I forgot to explain that those were serial transfers, the growth curves in the normal light-dark cycle for hundreds of days. The cells are growing rapidly without the stimulus, and then you give them the extended dark stimulus, and they still recover within 2 days. Unlike the naive culture, the dumb one, as I refer to it, that hasn't had the stimulus, it takes still takes 24 days to recover. So, does that answer your question? I can go back to the slide. Yeah. Oh, let me go.
Does it happen on signal transduction? Does it happen on signal transduction level or gene regulation level? Is that known?
No, we don't. We don't know anything actually. So okay. That's the I'm not on. That's okay. I can show you later that after we give them the stimulus of 3 days in the dark, and then it takes them 2 days to recover as opposed to 24. The learned ones. Then we put them back into their normal condition for hundreds of generations. They double every day, and we give them the stimulus, and they recover in 2 days. So but we don't know what the mechanism is. They've looked at methylation of the DNA, and there aren't any striking patterns there. So we're trying to go to a next step, some regulation or something.
But isn't it more of a selection process then? Rather than training?
Well, that's a good question, and we've sequenced the genomes, but you could say, is the sequencing deep enough to detect, some small changes? My genomics guys say it is. Say that the genomes are identical. We haven't selected for another strain. So, but that's a really good question and something we worry about. And we'll probably do the whole thing again and sequence deeper, and so it doesn't appear that there's been any selection for a different genotype. It's a mystery, though.
Okay, unfortunately, I think we're going to have to stop here. I could listen to her all morning long, but I think coffee is on the agenda next. We're back at 10:40, and let's give her another huge round of applause.